Welcome to Planted, finding your roots in STEM careers. Journey with us as we navigate the winding path into plant-related careers with professionals who have taken root or are still branching out. I'm Megan Wiesbrook.
And I'm Jessica Turner-Scoff. Today, we are just getting started on our journey, and we're talking about the somewhat surprising path that people take into plant professions. Our guest today is Dr. Andrew Hipp, and his own career path links together creative writing, the tree of life, and prairies. It's so nice to have you here, Andrew. How are things going, and how do these things come together?
Very well. First of all, thank you so much for having me. It's a real treat to be here talking with you guys. You ask how these things all come together. That's a great question. My grandma always used to ask me that. What is it that she always wanted to know if I was going to get a real job. How do they come together? I think of all of these coming together as they're all different ways of figuring out how the world is put together. In botany, I use a hand lens or a microscope. In writing, I use a pen. It seems to me to be all sort of the same thing.
Andrew, I've worked with you on a couple projects. You've assisted us with Teacher Institute Day and a number of programs in the past. It's always a pleasure to work with you. You have such great enthusiasm in front of any audience. And I know you're a senior scientist at the Morton Arboretum. What would you say is the coolest part of your job?
There's a lot of cool parts of my job. You talked about the teaching. I feel like it was really actually in some ways teaching that got me back into science. Because I like being with people and I like figuring out things. When I teach, whether it's for preschoolers or for graduate students or retirees or high school students, for me it's completely about figuring out how the world is put together and doing it with other people who are figuring it out at the same time. So one of the great things is the people that I get to work with, whether as a teacher, so I just talked about the teaching side, or being with people who have experienced a lot more than I have. So I've gotten to work with senior scientists, by which I mean people who are who've been doing this for a lot longer than I have, who are really patient and willing to convey their knowledge and spend time figuring stuff out together. In addition to the people, I love being in the field. This job gives me ample opportunities to collect plants, to tend a largish experimental garden that's completely overwhelming at times, very satisfying. And it gives me the chance to figure things out both by direct observation and by studying data and modeling data, figuring out things that I cannot observe directly about the evolutionary history of plants, at the same time that I'm making direct observations about how they coexist today.
So, Andrew, I know that you do a lot of work with our herbarium at the Morton Arboretum. Can you tell us a little bit about what a herbarium is, and also how does that compare to a living collection like the one you can see at the Morton Arboretum?
That's a great question. The herbarium is a collection of dried plants. It's a plant museum, which is the most boring description. I think functionally what it is it's a time capsule of plant biodiversity. People go out in the field, they collect plants that they observe in a given place at a given time. They press them so that they're dried. They label them so we know who collected them, where they went, enough information that I could drive back to that population 100 years hence. And something about the habitat, what it was growing with, what the soils were like. Then those plants are glued onto paper and they're stuck into the herbarium where they may last hundreds of years. The first herbaria were made in the 1500s and we still have some of those specimens around today. This stands in sharp contrast to a living collection, whereas we're trying to kill the plants and make sure that nothing's living on them so they'll persist forever. In the living collections, people are painstakingly collecting plants from China, from the Ozarks, from wherever it is that they're collecting, and getting those plants going in a common environment, often with a great deal of trouble, and keeping those plants going so that we can see what they grow like today. So creating a common garden, a garden where all these plants can grow together. The herbarium and living collections give very complementary views of plant biodiversity.
And so living collections can really support, they can be a gene bank for conservation, for making sure that these species will exist in the future. But can you talk a little bit about what type of research you can do with a herbarium specimen?
Yeah, herbarium specimens are interesting because they're physical objects and they are actually plants. We can go to herbarium specimens that are 100 years old or older, and we can get DNA out of them. so we can sequence them to understand what population diversity looked like, generations ago. We can go to plants that were collected in a given area and see what flowering dates were, what fruiting dates were, to see how flowering and fruiting have changed with changing climates. People have gone to herbarium specimens and looked at the density of stomata. These are the little breathing apparatus on the undersurfaces of the leaves, or the upper surfaces, depending on the species. The density of those stomata on the leaves correlates well with the amount of carbon dioxide in the air, in the atmosphere, so we can track changes in climates. In addition to that, people use herbarium specimens to track changes in the distribution of invasive species, changes even in, you know about some of the work that was done in ginseng.
That's right.
You want to talk about the ginseng study? It's a fascinating study.
They were able to use herbarium specimen and measure them over time and see that there was selectional pressure with harvest. Bigger plants were getting harvested. And so what was being found then was smaller and smaller plants over time.
This kind of work that dovetails with the research you did in your PhD work is best done from museum specimens that were collected broadly and opportunistically. What characterizes museum collections is that they are typically collected with an interest to documenting what's in a place, not necessarily driven by a hypothesis. When you have specific hypothesis-driven science, it often tends to be very targeted and efficient in its data collection. Museum collections tend to be rather inefficient in the sense that lots of stuff is being collected, but as a consequence, decades in the future, we'll be able to answer questions that the three of us sitting here can't even imagine right now.
Which I think is neat when looking at herbarium specimens, because they not only show that exact moment in time, it's kind of a preserved moment of what that plant looked like that day when it was collected, if it was flowering, if it had fruit, if there was any, fall color, if it is a specimen that has fall color. But the other thing that I think is so neat is that every time I look at them, there's so much information on when it was collected and where it was collected. And at first glance, there's the herbarium is huge. The number of specimens that we have at the Morton Arboretum is such a large bank. And so it seems like a lot of information to collect on each individual. But you bring up such a great point, is that information may seem like it may not matter right then, but can help, it opens the door to be able to look at it across a number of different species or across a number of different variables that you're trying to measure, because you don't necessarily know how you want to use it to answer what questions that may arise in the future. So such a cool point. And there's just so many neat things about herbarium specimens that I think get overlooked, but we obviously are all passionate about.
There'll be another show just on herbarium, so stay tuned.
Well, and that's such a nice transition, too, to talk about, not knowing where things are going to go in the future and the surprising path that your career has taken. Would you be able to just chat a little bit about your unique path into science and one that you initially hadn't planned?
When I was a young child, I wanted to be an astronaut. I loved space. I loved everything about planets and stars and spaceships. And I liked being outside. I liked horsing around outside. I liked to get up in the morning and go down to, there's a little creek by our house, the Menominee River, that we all swore was polluted enough that it would eat your feet off if you stuck your feet in. It probably wasn't that polluted. I don't even know. But it's like how I like to spend my time. But by the time I got into high school, I was really interested in writing and in the arts and in humanities. And so I had decided I'm not going to do science. I decided I'm not going to continue taking math, and I'm not going to continue taking science. I'm just going to focus on writing and literature. And that's what I did for a number of years. But because I liked being outside, I ended up taking, through a variety of paths, botany courses, with the suspicion that if I took enough natural history courses, I might get a job doing natural history. I thought that would be great. I'd like to spend more time being outdoors and maybe do it for a livelihood. And it ended up working out that way. And I ended up working as a naturalist and a ranger for several years. And in the course of doing that, discovered that all those years that I decided I wasn't going to study science, I had failed to learn a lot of science. And so I thought, well, okay, now I got excited. At this point, I'm 28 now, right? And I decided to go back to graduate school. It was really coming back to wanting to understand evolutionary biology, the evolutionary history of plants. And that's what brought me back to graduate school, was the stories that I was interested in as a writer, documenting, seeing how the world is put together. Then I wanted to understand that better. And so I went back to graduate school just to understand evolutionary biology.
Do you have a favorite type of writing that you enjoyed doing when it was kind of your major?
Oh, I was a very bad short story writer, and that's what I tried to do. And like everyone, when I was younger, I used to write poetry, also very badly, right? But I got my undergraduate degree as English with a creative writing emphasis, and my thesis was a little collection of short stories that I don't think is a very strong collection. But right after I finished, I realized that I liked essay writing. And so it's real, and that leads into all the writing that I do now. So it's really, really nonfiction.
Which I understand you're also a author of a series of children's books. Was that part of this major? Was that part of your career path or something else entirely?
Yeah, like so many things in my life, that was a tangent. That was a happy accident. Again, I had been a naturalist. I was back in graduate school now.
Okay.
And my wife, who had gotten her Master of Fine Arts in Madison, had a colleague, a friend, who was working at a publishing house. Her friend calls her and says, we have this set of 6 children's books on life history of different animals that needs to be written. Our author, who is going to do it, isn't able to do it. Any chance Andrew would be interested? And I said, yeah, that'd be great. I'd love to. It'd be natural history writing. I did a ton of research. I learned all these things about the animals, the earthworm and the snail and the mouse and I don't remember what else, and wrote these concise little books of about 1,000 words each. And it was just a ton of fun. But it really was not. It was not a part of my graduate work, but is it a part of my career? I feel like everything I do is part of who I am. So it's definitely a part of what I do professionally. Yeah.
And thinking about kind of adding to that skill set, over time, and building on it, what are some ways that you're able to use your both passion for writing as well as your skill set of writing well in your current role? So how have those skills aided you later on, maybe when, it happened haphazardly, you weren't planning on writing a series of children's book, but it's how has it helped you in your career now?
That one thing that I felt like I really learned as an English major and as doing creative writing was just this business of workshopping writing and working over prose over and over and over again. And it's helped me as a writer. You do a lot of writing as a scientist, whether it's grants or lectures or papers. And so I use my writing all the time in that way.
Andrew, it's just really clear that you've been able to combine your passions for writing, your compassion for the natural world, with research, and you've made your career your own. Now, do you have any advice for any young professionals or people that are just starting out who think that they're on a linear path? Because, as we know, there's rarely a linear path in this world. Do you have any advice you could share?
I agree wholeheartedly. I would say, first of all, try not to be scared about getting off of a set route. There's all these different ways of figuring out your life, and there's lots and lots of routes that seem very straightforward. They're often not, though. So very often people's careers take a winding road. So as long as that's the case, you might as well take charge of it. Mm-hmm. If you take a chance on what it is that you love, you risk probably very little, especially early on. Take the classes that most interest you, find what you care about, and dive all the way into it. Become the best at that. Whereas if you don't do that, if you take the classes that you feel you ought to, if you pursue the things that you think are going to be in your best interests, irrespective of your interests, you risk undermining your own happiness and not achieving as much as you could in the world. We're all here to do something uniquely, something that nobody can do besides Jessica, nobody can do besides Megan. And if you don't pursue that thing, you'll have missed your shot.
And I think it just really speaks to the fact that, you didn't know that you were going to be a senior scientist someday. And you kept making choices based on your interests and building your skill set and your building opportunities based on the things that interested you. And I know with myself, I did not know that I would be a science communicator someday. I had a thought that I was going to be very linear. I was going to be in academia. I was going to be teaching at a small college. But I realized that, in college, I did a lot of theater and different sorts of speech and a radio program. But it's, those skills really built upon themselves, and they've allowed me to make my own career. So, and Megan, I know you have the similar story as well.
Yeah, I mean, I thought I was going to be a marine biologist, and then got a little bogged down and didn't want to be as much on a straight biology path, but really loved working with kids. On a whim, I took an education class and ended up in a fifth grade classroom and loved it, and kind of switched my major overnight to education with a focus in science, and then taught middle school and loved that, but missed being outside, missed having the opportunity to really inspire kids about science. And so looked for different types of teaching and different types of environment, and found myself at the Morton Arboretum. So yeah, different.
Grateful you did.
Yeah.
You're great.
I'm happy to be there. I know a lot of your kind of making this career of your own and betting on yourself. One of the things you mentioned that has been the coolest part of your job is getting to meet so many different people and talking with them about your work and teaching them about your work, or also learning from others who have come before you and other senior scientists, of that being kind of the cool part of your job. And this is a loaded question, so it might be kind of hard to answer. But what has been the coolest experiences or most amazing things that you've gotten to do along the way? A cool project, a really interesting thing that you want to share?
I feel like so many projects are microcosms of everything you do. And I guess our work on the Oaks has been a microcosm, I think, of everything, because it's involved so many different things. I got into Oaks only because I got a job at the Arboretum. So it was a happy... No, I'm serious. I was a sedge guy. I was interested in sedge biodiversity, and yet I got a job at an Arboretum, and I knew it was a good fit, and I knew that I had to work on woody plants because that's the mission of the Arboretum. So I thought, well, I'll work on oaks. So here you got the happy accident. So that's been great. Then I got interested in this one problem, which is just the Hills oak, this cool Chicago endemic. Understanding that sort of cracked open an invitation from a senior colleague, Paul Manos, to work on the oak tree of life. And Paul's brilliant idea was that we can do this. We can figure out the oak evolutionary history. He was right, and so we got this whole group of scientists. So I got the sort of to come into the fold with Paul and all these other scientists who are doing work that I didn't even know about. And coming out of that has been opportunities to work with schools where we do work trying to understand just sort of oak distributions at the local scale. With colleagues in Mexico, I've been sort of gotten to do field work with colleagues in Durango who've been extremely inviting and kind, and colleagues in Morelia. And then discovering new analytical tools that I had no idea were out there, all because of oaks. Thank you, oaks.
And I don't know if you can break apart or break down exactly what you mean by oak tree of life. I know that this is a large subset of data, that is a lot of different types of data that you're putting together to kind of essentially tell a story. But can you break that out as to what exactly that project entails and what you're looking at as part of that project?
Yeah, let me try. This is the field of I am going to try, honestly. This is the field of systematics. It's trying to understand from data that we have today, all these pinpoints, sort of imagine that you've got a black curtain, right? And all you've got is these little individual plants poking out of it. And you know that behind there, this is history of all these populations leading to each other, generation after generation after generation, leading to the individuals that you collected. But back in time, somewhere behind that curtain, those populations come together 10 million years ago, 20 million years ago, 30 million years ago. That's evolutionary history, right? And so as a systematist, as a plant taxonomist, my job is to take these moments at the tip and infer what's going on behind the black curtain. So how do we do that? It's so amazing that we can. What we do is we chop up the genome, right, into all these sections where we have information at individual regions of the genome about what individuals are related to which individuals. We then search amongst all the possible sets of relationships amongst those species and say, what set of relationships would maximize the probability of the data we observe today, these gene sequences, the A's, the C's, the T's, the G's, if we understand how mutation occurs. We run through bajillions of trees. It turns out that if you have 70 tips on your tree, just 70 species, the number of possible trees is somewhere near the number of elementary particles in the universe. So there's tons of trees you have to run through. Fortunately, we don't have to do that by hand. The computer will run through them, and people have gotten very clever. Programmers are very clever about figuring out how to search amongst this huge tree of life space, the space of possible trees of life. Once we've done all that, we get a model inside the computer, a sort of a statistical picture of the tree of life, and then it's as simple as literally drawing it, where the lengths of the branches correlate with numbers of millions of years from this species back to its common ancestor with this species over here. And so what you get is a drawing that you can look at, and you can hang data off of it. You can look at how ecological conditions have changed over time and how the attributes of the plants that fit them to those ecological conditions, how they're adapted, how those have changed over time. And ask what's happened in the past as a window to what may happen in the future.
So what makes the oaks such a tricky group to work with? Because I know that there's close to 450 species. Why has this been a question that hasn't been answered yet?
Part of it has been, a little of it has been our perception that oaks are utterly promiscuous, that any oak species will mate with any other oak species. And that's kind of true. They do hybridize at a low rate. Probably the bigger issue, though, is that in addition to this rate of hybridization, which is going on all the time, right? But in addition to that, oaks have these great big populations. So when an ancestral species splits into two, each of those daughter species inherits a lot of the gene diversity that was in the parent species. That process that we would like to record, that instant when species diverge, there isn't probably an instant. And it's not like a single population evenly dividing up the genes. Okay, you get this gene copy, you get this gene copy, go play separately now. It doesn't work like that. So as a consequence of those two phenomena, you need lots and lots and lots of genetic data. And even then, there's parts of the tree of life that dog us even now that we're not going to get figured out, perhaps in my lifetime. But we can get a lot figured out. We've gotten a lot figured out so far.
It sounds like the more pieces that get put together, almost the more questions it creates, more things that you want to learn more about the species as more of the pieces come together, which I think is a natural part of the process of being a scientist and discovering more and wanting to know more at the same time.
It really is, this whole notion that there is a scientific process. I don't think there is a scientific process, but there is a scientific habit. It's this business of observing the data, trying to fit it to a suite of hypotheses to those data, and then saying, okay, given what I figured out now, what more do I want to know? And going and grabbing more data and setting a new set of hypotheses you want to test. Yeah.
We recently had teachers on site at the Morton Arboretum for a workshop day, and we showed them your profile, and all of our guests actually, and asked them what type of questions that they'd want answered from you. So we can really, make sure that this podcast has a lot of value and meets the needs of our audience. One of the questions that these teachers wanted to know is, what does your day-to-day look like? And I know that you have field, you have lab time, so there's probably no typical day as a scientist, but maybe if you could talk a little bit about field versus lab days, what that is.
Sure. We have different kinds of field work that we do in my discipline. We have projects. I have projects where I'm out collecting plants. So in that case, I and collaborators or I alone will be driving across the landscape, looking for populations or going to known populations, collecting plants that we can then bring back to the herbarium. So we have a solid record of what we've collected, and into the lab so that we can grind them up and get DNA out of them. And that's a lot of discovery. That's a lot of on the ground, whether it's in the Rocky Mountains or in Mexico or in the Midwest. Other kinds of fieldwork we do. We have an experiment that we're doing on the west side of the Arboretum where we're looking at the effects of biodiversity on prairie restoration. It's a giant garden that every 2 meter by 2 meter prairie that we're planting out there needs to be tended. So there are days that we're just weeding. I would have to say, though, a lot of my time is spent analyzing data and writing papers to keep making sense of the data that we're bringing in and being able to understand what direction to go next. As Megan put it very nicely, the questions that we're asking now bring up new questions, and they always suggest a next direction to go. And it's my job as a primary investigator for our lab is to keep that axis going forward. And I love doing that. It's a ton of fun.
Andrew, we've so enjoyed talking with you today. There's so much passion and everything that you have to say. To some extent, I wish some of our listeners could see, as you were describing the Oak Tree of Life, how you were putting it together visually as well, so that we could see you drawing those connections.
Oh, there's been miming, there's been drawing. It's been awesome.
But we do have one last question for you, and I know that this is a tough one to choose, but we will make you pick just one. What is your favorite plant?
I'm going to go with bladderwort.
Okay.
And so bladderworts are these aquatic, well, I know them as aquatic plants. There's also some that are found in wetlands, but the ones that I became most familiar with early on are submerged plants. They have filamentous leaves, and so you'll find them in a lake. They look rather unremarkable until you look closely and find that they're covered with these little sac-like structures, about 1 millimeter, 2 millimeters across or smaller, called bladders. They were not known for a long time what their purpose was. In the late 1800s, folks thought maybe these are involved in flotation, but they clipped off the bladders and found that the plant still floated all right. In some cases, they thought that the little hairs on them were involved in absorbing nutrients, and the bladders were to shield them from the sun. I don't know how they ruled that out, but in the 1870s, there was a botanist who went into the herbarium and clipped open the bladders on all these herbarium sheets and found little crustacean skeletons in there. And it turns out that the bladders are a tool for the plants to gobble up little invertebrates that go floating by the plant, and little hairs on the base of the bladder are like triggers. They trigger those little hairs. The trap door in the bladder snaps open. The crustacean gets sucked in, and the trap door shuts, and the crustacean can swim around happily and slowly being digested. And so the plant devours its prey in this way, very slowly, very calmly, but heartlessly all the same. So yeah, I like bladderworts.
And a great example of how herbarium specimens can be used to answer questions you didn't know you had.
Exactly.
Thank you so much for joining us today, Andrew. It's been just a delight hearing about your experiences and how you've combined your passions and forged your path in a non-linear way to being a senior scientist, and it's just been great talking to you.
Well, thank you so much for having me. I've thoroughly enjoyed this as well. It's a real honor to be asked.
This episode of Planted, Finding Your Roots in STEM Careers, was made possible by the Morton Arboretum and Multimedia Services at College of DuPage. Check out additional resources and bonus content at www.mortonarb.org/plantedpodcast. This is Jessica Turner-Scoff
and Megan Wiesbrook,
and we hope you'll continue to join us as we journey into STEM careers.